Okay, so protein synthesis so in protein synthesis there are these things called codons, and these are a sequence of three DNA or RNA nucleotides that correspond to a specific amino acid, or a stop codon, or even a start codon during protein synthesis. There are 61 different codons, 3 are stop codons, which stop transcription, and then one start, which kind of gets the whole process going. So those 3 different stop codons are UAA, UGA, and UAG. And then the only start codon is MET, or MET. And as you can see here, here's an example little transcription thing. Here's the amino acids. Here's the stop, here's a start, and these are just the 3 different codons. Okay, so for transcription, that's the first step in gene expression. DNA unwinds and creates the transcription bubble, which is seen right here. One strand is used as a template strand. RNA polymerase transcribes DNA into RNA 5' to 3' direction. And there's 3 different complexes. There's RNA polymerase 1, 2, and 3. For eukaryotes, the initiation promoter is called the TATA box. And it's seen right here in this picture right here. It's a sequence of, I'm just going to say letters that essentially spell out TATA. Hence why it's called the TATA box. Okay. Now for translation, translation is the next part. It's read from 5' to 3' by the tRNA, or transfer RNA. Initiation is the first step, and this is when the ribosome gets with the mRNA and first tRNA to begin translation. So it's seen right here. It's coming in right here. Complex of small ribosomal subunit, and initiator tRNA binds to the 5' cap. Here we go. It's binding right there. Elongation. Amino acids are brought to the ribosome by tRNA, transfer RNA, and links them together to form a chain. And then that is the elongation process. And as you can see, so what's going to happen is there's going to be a long chain of proteins that are essentially just going to start forming behind the MET. And the termination is the last stage. It's the finished polypeptide is released in the cell, and he goes to work and gets the job done. Okay, so breaking this picture down right here, the complex of small RNA ribosomal subunit and initiator tRNA binds to the 5' cap. So here it is, the initiator tRNA and the small ribosomal subunit is coming in right here to bind. And then it's going to scroll over to the start codon. And it scans to find the start codon. So it's kind of like a barcode scanner thingy. And then the initiator tRNA binds to the start codon. So he's ready to start doing some transcription. And then the large ribosomal subunit will come. And then remember those 3 complexes? It has these things in there called EPA. And these are essentially the different spots where the little polypeptide chain is going to get read into. So what happens is this will essentially move from here to here. So this is where they'll bind, right here. And then it's going to get moved here to here, and then it's going to go away. So a quick overview of that, a quick overview of everything. You have transcription, which is where the RNA is essentially coded. The RNA that it's going to, the tRNA is going to read and everything in the ribosomal subunit is coded in the transcription process using the transcription bubble. So here's our regular DNA. Okay, so now it's being transcribed into its correlating information, the RNA. The RNA strand is being created. And that information is going to go off and then start translation, where it's going to turn into the polypeptides and get read and turned into these, the polypeptides. And here are the codons. And it's always 3 separate ones. And that is a quick, rough overview of protein synthesis, transcription, and translation.